Hallo alle sammen. Da tror jeg vi kan komme I gang igjen. Det meste av det som skal skje nå, det skal skje på engelsk, men når jeg ber dere være stille, så kan jeg gjøre det på norsk. So, welcome everybody to the second, or rather third part of this day's conference in the field of epigenetics. We are now switching to English because we have our guest star, today's main star, Stephen Suomi from, and here I have to take my glasses and the sheet of paper, or else I wouldn't be able to remember this whole title. Stephen Suomi is chief of the Laboratory of Comparative Etiology at the Eunice Kennedy Shriver National Institute of Child Health and Human Development in Maryland. And Stephen is a trained psychologist, but he's one of those people who has had courage enough to enter into real interdisciplinary work. He is famous for his work in epigenetics on monkeys, and that's what he's going to tell us about today. I saw Stephen a bit less than a year ago in Washington, and I heard him, and I saw his monkeys, or not the monkeys, but the pictures. And you're really up for a good time, I can tell you. This is really, really thrilling what he has to tell us. So this part of the conference is was sort of made up or planned mainly through the seminar in science studies. And I went last year on behalf of the seminar to Washington to something called the AAAS. I don't know if everybody here knows what that is, but it's the American Association for the Advancement of Science. And Stephen Suomi was there. And not only does Stephen have the courage to cross between disciplines and to do interdisciplinary work, he also has the courage to have an engagement in the public reception of science, and not only, of course, on the receptive sides, but on that side he gives science to a larger and broader public, which is really admirable. Stephen also told me yesterday that he's engaged in another issue, and that is the issue of the relationship between science and politics. And he has firm conviction that politics can be better if their decisions are based on not only evidence, but presumably on something we can call knowledge. But today, Stephen is not going to talk about politics. He is going to talk about his research on these fascinating rhesus monkeys and its relationship to epigenetics. So please, Stephen, come up here. The floor is yours. You just talk, and it will go directly there.
Everybody hear me? Thank you, Camilla, for that lovely introduction. And it is a pleasure to be here in Oslo for the first time, hopefully not the last, and have the opportunity to tell you about the work some of my colleagues and I have been doing in my lab at the National Institutes of Health. My lab is not on the main NIH campus in Bethesda, Maryland, where most of the science is performed, but rather out in the Maryland countryside, about 40 kilometers up the Potomac River, near a little town called Poolesville. So when I talk about the Poolesville colony, that's what I'm referring to. And there, in basically farmland and forest land, we have the opportunity to have 400 rhesus monkeys living in naturalistic most of them living in naturalistic situations. And my lab, from its inception, has been interested in the study of individual differences in the personality or the temperament of these monkeys and the biological substrates that underlie these individual differences, and how genetic and environmental factors act, and we think actually much more importantly, actually interact to shape individual developmental trajectories. And over the years, we've been especially interested in two subgroups of monkeys, not only at our Poolesville colony, but also at two field sites that we've had the opportunity to have access to over the years. The first subgroup, comprising about 20% of the population at Poolesville, and also about 20% of the monkeys in the wild, are individuals who seem to be unusually fearful or anxious in the face of novel or mildly challenging circumstances. So situations and stimuli that most monkeys find really interesting and will readily explore, these monkeys try to avoid. They're also shy in new social situations. And when they're forced to confront these physical or social situations, they show profound arousal of a variety of biological systems. The system we've paid the most attention to over the years is the Hypothalamic-Pituitary-Adrenal Axis, or the stress hormone system. And we measure this most often in terms of cortisol, whether the glucocorticoid cortisol, whether we're collecting it in blood or saliva, or more recently in hair. These monkeys and these individuals tend to have, under challenged circumstances, much higher elevations of cortisol than other monkeys, and they stay up for longer periods than these other monkeys. And this subgroup of fearful, anxious monkeys, we call them our uptight monkeys, look very much like a subgroup of human children that people like Jerome Kagan at Harvard and my friend Nathan Fox at University of Maryland and many others have been studying for the past quarter century. These are children that Kagan calls behaviorally inhibited. They show the same reticence in novel situations that our monkeys do, and the same biological changes. And we now know that these children are at risk for developing childhood anxiety and depressive disorders, and for some of whom this risk carries on through adolescence and into adulthood. There's a second subgroup of monkeys that we focus on, comprising maybe 5 to 10% of the population at Poolesville and 5 to 10% of the population in the wild. These are monkeys who seem to be unusually impulsive, and their impulsivity gets them into trouble. They do stupid things that other monkeys know better than to try, like get between a high-ranking female and her offspring, or they may repeatedly confront a dominant adult male, and when that male beats them back, they're right back in that male's face. And when that male beats them back again, they're right back in that male's face. So not surprisingly, they tend to elicit a lot of aggression from other monkeys in the group, and they themselves are excessively aggressive, and more importantly, once they get into an aggressive encounter, whereas most monkeys know how to back off and cool things off before things get out of hand. When these guys are involved, the intensity of the aggression tends to escalate to the point where somebody may actually get hurt. These monkeys also have biological features that differentiate them from others in their group. The feature we've paid the most attention to is an apparent deficit in serotonin metabolism that we measure primarily in terms of chronically low levels of the primary central serotonin metabolite in cerebrospinal fluid, 5-hydroxyindoleacetic acid, or 5-HIAA. And monkeys who are chronically aggressive tend to have who are excessively aggressive tend to have chronically low levels of this serotonin metabolite. And these monkeys also resemble a subgroup of human children that people like Dan Ulvanis here in this country and my friend Richard Tremblay in Montreal have been studying for years. These are children who are already characterized as being excessively aggressive by the time they're 2 years of age, and whereas aggression in most kids drops with time, it stays high in these kids. By the time they hit the school system, they're often causing classroom disruptions. They're often diagnosed as having externalizing problems. And in my country, at least, many of these kids, by the time they're in their mid-teens, you can find either in prison or in the morgue. So we've been interested in these two subgroups, not only because they're apparent resemblance to subgroups of human children that are of considerable interest to both clinicians and parents and policymakers, but also because in both cases the behavioral and biological characteristics that distinguish them from others show up very early in life, and in the absence of major environmental change are remarkably stable from infancy through the childhood years, adolescence, and even into adulthood. And we know that some of these characteristics are highly heritable. That is, there's an obvious genetic component to the differences that we see. So, for example, in our uptight monkeys, the high levels of cortisol are highly heritable. And for the impulsive aggressive monkeys, individual differences in 5-HIAA concentrations are also highly heritable. But I'm here to tell you this afternoon that those characteristics, even highly heritable biological characteristics can be modified by experience, particularly early social experience with caregivers. And this modification with experience shows up not only at the level of behavioral expression and emotional regulation, but also at the level of neuroendocrine function, at the level of neurotransmitter metabolism, at the level of brain structure and function, and we now are finding out even at the level of gene expression. So what I want to do today is to flesh this story out. I'm going to begin by telling you a little bit about the monkeys that I study, because they are a very unusual species of primates, unique in many ways. Then I'm going to describe how they grow up under normal circumstances, what happens when those early experiences, social experiences, are modified, talk about gene by environment interaction, present some recent data looking at very early mother-infant interactions, because we think now that some very important things are happening in those initial days and weeks of life. And then finally, time permitting, put all of this into a comparative perspective. So let's start off with rhesus monkeys. Rhesus monkeys are a member of the macaque genus. There are 20 or 21 species of macaques, depending on who your taxonomist is. Rhesus monkeys are one of them. And they are clearly not our closest relatives from an evolutionary standpoint. They only share about 95% of the genes that we have. By contrast, or in comparison, chimpanzees and bonobos, our closest evolutionary relatives, share between 98 and 99% of our genes. But rhesus monkeys are a lot more like us than our chimpanzees or bonobos or any other of the great apes in one fundamental respect. They are a success story. They are the world's second most successful species of primates next to us. There are more rhesus monkeys living in the wild, encompassing a wider geographic range, experiencing a wider range of climates than any other species of non-human primates. And in the Indian subcontinent where they come from, you can find them everywhere from rainforest to hardwood forest, to coniferous forest, to savanna areas, to the edges of the desert, and even into the foothills of the Himalayas, where the winters are every bit as severe as what you have around here. So we have no problem maintaining a colony of rhesus monkeys outdoors year-round in rural Maryland with minimal shelter. Secondly, they're one of the few primate species, we're one of the other ones, that can be genuinely called a weed species. That is, if you take them out of their original habitat and put them someplace else, they're likely to thrive and expand their population, whereas most other primates would readily die off. So every time somebody's taken rhesus monkeys out of India and put them someplace else, whether it's an island off the coast of Puerto Rico or the swampland outside of New Orleans or the high arid plains of western Texas or the Sacramento Valley in California, where it gets to be 40 degrees centigrade every day during the summer, or rural Maryland, these monkeys do just fine. And finally, they're remarkably resilient, and I can give you many examples, but I'll just give you one. I don't know how many of you in this audience remember Hurricane Katrina, this massive storm that demolished New Orleans and the Mississippi Gulf Coast about 6 summers ago. What you may not know is 40 kilometers north of New Orleans on the northern shore of the Lake Pontchartrain is the Tulane National Primate Research Center, where they had 3,000 rhesus monkeys living in outdoor pens when the hurricane hit. And when Katrina went through, it caused enormous damage, not only to New Orleans, but also to the Primate Center, essentially blowing away all of those pens. Yet every single one of those 3,000 monkeys survived. They didn't lose a single one. And we think we know how that happened. And each of those pens was a concrete culvert, the kind of things that they use for drainage under expressways. The caretakers put them in there so that the monkeys would have a place to get some shade from the hot Louisiana sun. And when the hurricane hit, almost certainly what happened to these monkeys went into those culverts, rode out the storm, watched the rest of the pens blow away while they were in the culverts, and then waited for the humans to show up for their next meal. So these monkeys handled Hurricane Katrina a lot better than most of the people in the New Orleans area. In the wild and in those captive environments that so provide, these monkeys live in large social groups called troops. The troops can range in size from 30 or 40 individuals on the small end to over several hundred on the large end. But whether the troops are large or small, they all have the same basic social structure. Every troop is organized around several female-headed, multi-generational families, or matrilines, plus males who have come in from the outside. And this arrangement derives from the fact that rhesus monkey females stay in the group in which they're born for their entire life, whereas rhesus monkey males stay only until puberty, and then they leave. They typically then join all-male gangs, hang out in those gangs for anywhere from several months to a year, and then try to work their way into a new troop. So every troop has plenty of adult males and females in it, but the females have all been there since birth, and they're all related to one another, at least within their respective families, whereas the males have been there only since puberty, and initially they're related to no one until they start having offspring of their own. And here's one typical rhesus monkey family. Here's a mother, newborn infant, grandmother's back there. This is an older sister of the infant. This is a younger sister of the mother. The other thing you should know about rhesus monkey social organization is within every troop there are multiple dominance hierarchies. There's a hierarchy, for example, between families, such that every member of the highest ranking family, including infants, outranks everybody in the next highest family, including adults, who outrank everybody in the third family, etc. So you often see situations in the wild where youngsters, infants from high-ranking troops may be stumbling along a trail, and adults from lower-ranking troops will be falling all over themselves to get out of the way of these little infants, because they know if they cause those infants any grief, the rest of that high-ranking family will be on their back in no time at all. There's a separate hierarchy within each family that follows a general rule: younger daughters outrank older daughters. And it's easy to see how this happens because the mother will preferentially defend her newborn daughter from the harassment of a jealous older sister. So even though the older sister is bigger and stronger than her younger sister, with the mother's backing, that younger sister can get away with anything she wants in those interactions. The interesting thing is that this relationship among sisters is maintained as long as both sisters are alive, even after the mother is no longer around to selectively reinforce these relationships, because where the mother leaves off, other members of the troop take over and continue to give deference to the younger sister in her interactions with her older sister. There's a separate hierarchy among the males that come into a troop that's roughly related to tenure. That is, the longer a male is in a troop, the more likely that male is to be high-ranking. But of course, it's not tenure or length of time that's important. What's really important is how good that male is at making friends and sustaining alliances, not only with other males, but especially with high-ranking females who carry the real clout in the troop. And males that are good at that move up their hierarchy, and males that are not so good give up, leave, and seek their fortune elsewhere. And finally, there's a hierarchy among the infants that are born into the troop every year that basically reflects the position in the troop of their mother. So it's not that some infants are bigger or stronger or smarter or quicker than other infants. Those differences undoubtedly exist. It's that other monkeys in the group associate a particular infant with a particular mother and afford it equivalent social status. So life in a rhesus monkey troop is a pretty complicated business. For an individual to survive, let alone thrive in this group, not only must it establish a complicated social repertoire of its own, but it also has to acquire a great deal of knowledge about other members of the group. Who's related to whom, where does each individual fit in these different dominant hierarchies, as well as recent social history. Who's been getting into fights recently? Who can you count on to back you up if you get into a fight yourself? And monkeys that are able to assimilate all this information and follow the rules inherent in these multiple dominance hierarchies do very well in these complex social groups. And monkeys who are either unable or unwilling to follow these rules don't survive very long at all. So how does this socialization process work? Rhesus monkey infants, who by the way grow up 4 times faster than we do, which is an enormous advantage for those of us who are interested in studying long-term effects of early experience or lifespan development or the transmission of characteristics from one generation to the next. This 4-to-1 ratio is useful because we can see a generation in 4 or 5 years instead of having to wait 15 or 20 for the human equivalent. So when I give you an age in rhesus monkey months or years, just multiply by 4 and you get a rough human equivalent. At any rate, rhesus monkey infants spend virtually all of their first month of life in intimate physical contact with their mother, usually in this ventral position, or at no more than arm's length away from the mother. And during this time, a strong and enduring attachment bond is established between mother and infant. It's a bond fundamentally equivalent to the kind of attachment relationship that human infants routinely form with their caregivers in every culture in the world. And while this attachment is being formed, mothers are very picky about who they let interact with their infant. They usually don't let adult males anywhere near their infant, which is one reason why in this species, not in all primate species, but in this species in particular, adult males have very little to do with the early child rearing. These females usually don't let unrelated females get too close either, and this particular female is not about to let the photographer get one step closer. These infants don't stay infants forever, however, and beginning in the second month, they start leaving their mothers for short periods of time, and they do this in a very stereotypic fashion. They'll leave their mothers for a few seconds, go a few meters away, then go running back to their mothers for a period of contact, go out and explore a little bit more, and then running back to their mothers for a period of contact. It's almost as if the infant is attached to its mother by a long rubber ball. And as the weeks and months go by, these exploratory forays become more frequent, involve greater distances away from the mother, and longer periods of time away from the mother, so that by the time they're 6 months of age, they're only spending about 20% of their waking hours in actual physical contact with their mother, as opposed to the 100% that they were spending in their first month. Yet the presence of the mother is absolutely crucial to sustain this early exploration, because if you lose access to your secure base, as John Bowlby has taught us in his human attachment theory, all motivation to explore disappears and you rapidly become upset. So who are these young? So the mother remains an important part of the infant's social world, even though the infant isn't actually interacting with her that much. So who do these youngsters spend time with? Well, they spend time with peers. And this is not by accident. In the wild, Rhesus monkeys are seasonal breeders. They copulate year-round, but the actual conceptions only occur within a 2 to 3 month period every year. So what this means is 80 or 90% of a troop's infants are born within a 2 to 3 month window every year. And what this guarantees is that any youngster born during this time will have plenty of other youngsters who are about the same age and about the same physical, cognitive, and social skills as they have. And that's who they choose to hang out with. So these monkeys, throughout the childhood years, the rest of the first year of life, the second year of life, and the third year of life, will spend many hours every day in active social play with their peers. And it's in the context of this play that virtually every behavior pattern that will be important for normal adult functioning can be developed and practiced and perfected long before it actually has to become functional. And work by my mentor Harlow way back in the 60s, as well as much more recent work by Dutch investigators, have convincingly demonstrated that Rhesus monkey youngsters who are otherwise well socialized but denied the opportunity to play inevitably grow up to have problems with reproduction and real problems controlling their aggression. And then when these monkeys hit puberty, which is the beginning of the third year, end of the third year for Rhesus monkey females, when they have their first menses and thereafter have regular 26 to 28-day menstrual cycles, and males the beginning of the fourth year when their testes enlarge and descend and both genders show pronounced growth spurt, that's when the genders go their separate ways. As I mentioned, Rhesus monkey females stay in the group they're born for the rest of their lives, and they stay fairly close to their mother. This is a mother-daughter pair from one of our outdoor groups at Poolsville. The daughter is on the right, and 6 months after the slide was taken, the daughter became a mother for the first time, and that's where you see the real strength of the matriline, because the birth of a new infant, especially to a new mother, has the effect of bringing the rest of the family around to buffer that new mother and infant from outside social stress. And then females will continue to play an active role in family life even after they're no longer having kids of their own. So grandmothers and great-grandmothers are just as important cogs in that family as our mothers and infants. The story for males is completely different. Males, when they hit puberty, they leave. They either leave voluntarily or they're physically kicked out of the group, usually by unrelated females. And then these males go and join all-male gangs. They'll hang out in the gangs for anywhere from several months to over a year. And then they'll try to work their way into a new troop. And we now know two essential things about this male emigration. First, without a doubt, it's the most dangerous period in the life of these males. There's nothing like it prior to puberty, there's nothing like it after puberty, and there's nothing comparable for females. It's been estimated that the mortality rate for these males from the time they leave home until the time they're successfully integrated into a new troop is between 40 and 50%. It is a very rough world out there for those young males, and almost half of them don't make it. We also know that there are big individual differences among these males in both the timing of emigration, when they leave, how old are they when they leave, and the strategy that they use to get into a new troop. And I won't go into the details of this, except to say that we're getting pretty good at predicting when a particular male is going to leave home and what he's going to do there afterwards based on the behavioral and biological profile that he shows us while we're growing up. So this is a sequence that Rhesus monkeys go through from birth to maturity, and they've been doing it for hundreds of thousands, if not millions of years and generations. And I fully expect them to keep doing it for just as long, if not longer. Indeed, I think they're going to be around as long as we are, because they can live just about any place that we can and some places that we can't. But then again, I really like Rhesus monkeys. Now, not all Rhesus monkeys at Poolsville grow up this way. Some are cross-fostered at birth to unrelated females, so we can separate out genetic and biological factors. And some are reared according to a procedure developed by Harlow many years ago without parents, what he calls together or peer-only rearing. Here, monkeys are separated from their biological mother at birth long before any kind of an attachment relationship can be established. They're hand-reared in a neonatal nursery by human caregivers, and then within the first month of life, they're put into small groups of 4 or 5 individuals like themselves, where they will spend the next 6 months with one another 24 hours a day. And then at 6 or 7 months, these monkeys are moved into larger social groups containing their mother-reared peers, because at this point, that's the age in which, in the wild, rhesus monkey mothers wean their infants. So from 7 months on, mother-reared and peer-reared monkeys are living in exactly the same physical and social environment. So any differences you see in them subsequently can be attributed to their differential social experience during that first 6 months of life. And when monkeys grow up this way, they become hyper-attached to one another, much stronger attachments than you'd see between a mother and an infant. But these attachments, while very, very strong, are actually dysfunctional, because what happens is these infants spend excessive amounts of time clinging to one another, and they don't get the opportunity to go out and explore their environment to the extent their mother-reared counterparts would do. And when they get to the age where they should be starting to play, they do indeed start to develop patterns of play, but the patterns never reach the level of complexity and intensity and frequency that you see in their mother-reared counterparts. And when you think about it, it's not surprising, because these monkeys have to serve as playmates and as attachment objects at the same time, and they end up doing not so great a job of both. So even though these monkeys are with potential playmates 24 hours a day, they actually are play deprived as they are growing up. And this has its consequences. For one thing, peer-reared monkeys later in life appear to be more fearful than you would expect to see from their mother-reared counterparts. And in this respect, they look behaviorally exactly like that 20% of monkeys in the wild who show excessive fear and reactivity to new situations. And like the 20% of monkeys in the wild, they show higher levels of cortisol under comparable challenge circumstances. Something else happens to these peer-reared monkeys as they get older. Not only do they become more fearful, they become more aggressive. And they look like that 5 to 10% of monkeys in the wild who seem to have excessive amounts of aggression and in an escalating fashion. And like that 5 to 10% of monkeys in the wild, they show deficits in serotonin metabolism, whether you're looking at the first year of life when 5-HIAA levels are still pretty high, but there's a significant difference, or year 5, this should be 5, not 2, late adolescence, early adulthood, when the levels have dropped for everybody, but if anything, the rearing condition difference is bigger than it was before. And even though I said differences in 5-HIAA are highly heritable, these differences are not the product of genetic differences, they're the product of differential early experience. When some of our monkeys get to be adolescents and young adults, my colleagues in the Alcohol Institute, National Institute of Alcoholism and Alcohol Abuse, run them through the monkey version of a happy hour, where for every day for many weeks or several months, these monkeys have unlimited access to 9% NutraSweet flavored alcohol, or non-alcoholic NutraSweet flavored beverage, or plain tap water. So they're not fluid deprived, and they consume alcohol in familiar situations. Under these circumstances, some monkeys consume more alcohol than do others, and as a group, peer-reared monkeys consume significantly more alcohol than their mother-reared counterparts. More recently, we've been able to do neuroimaging studies of these monkeys when they're a little bit older, PET scans and structural MRIs. Here are images from a PET scan study of monkeys who are now 2 years of age. They've been living together for 18 months, but differed in their rearing for the first 6 months of life. And what you see on the left is serotonin binding potential, on the right is cerebral blood flow, and the comparisons are between a composite mother-reared versus peer-reared from the front, composite mother-reared versus peer-reared from the top, composite mother-reared versus peer-reared from the side, and similarly mother versus peer, mother versus peer, mother versus peer for cerebral blood flow. And simple inspection shows you that the peer-reared monkeys' brains are not lighting up as much as their mother-reared counterparts. And when you look at the actual data, that's indeed the case. Throughout the brain, whether you're looking at the raphe, the thalamus, the striatum, the temporal, or the frontal regions, peer-reared monkeys consistently have lower serotonin binding potential than their mother-reared counterparts. And the same is true for the cerebral blood flow. I won't show you a picture, but it's about the same. We recently published a paper looking at serotonin 1A receptors in a PET study. You get the same pattern of differences throughout the brain. We published recently also a functional MRI study, a structural MRI study looking at structural differences in the brains between mother and peer-reared monkeys, and the brains are different. So early peer-rearing not only has the effect at the level of behavioral expression and emotional regulation, more fear, more aggression, at the level of neuroendocrine function, higher levels of cortisol, at the level of neurotransmitter metabolism, lower levels of 5-HIAA, but even in terms of brain structure and function. And very recently, here's the epigenetics part in collaboration with Moshe Ziff at McGill, we've been able to look at genome-wide scans of methylation patterns in differentially reared monkeys. And what this figure shows, chromosome by chromosome, is genes that are either more methylated in mother-reared, if they're above the line. Each of these bars is an individual gene, and if it's above the line, it's more methylated in mother-reared monkeys. If it's below the line, it's significantly more methylated in peer-reared monkeys. And this is prefrontal tissue from prefrontal cortex when the animals are adults. And these are lymphocytes collected at the same time when the animals are adults. And here's chromosome 2, here's chromosome X. And what you can see is there are an awful lot of genes that are differentially methylated as a function of that first 6 months of life, even at 7.5 years later. In fact, fully 1/5 of the entire Rhesus monkey genome, over 4,400 genes, are differentially methylated as a function of that first 6 months of life. And furthermore, even though there is some tissue specificity, there are some of these genes that show exactly the same pattern, whether you're looking in the brain or in the blood. So these genes on chromosome 1, for example, are all being significantly more methylated in mother-reared monkeys than in peer-reared monkeys, both in brain and blood. In this case, it's just the opposite, and about 30% of those 4,400 genes show this pattern. Here, you see the opposite pattern. If it's more methylated in the brain in mother-reared monkeys, it's more methylated in the blood in peer-reared monkeys, and here and in here. So, and that's about 25% of those 4,400 genes. So even though there is some tissue specificity, there are also a lot of genes that are being shown methylated in the same direction in both sets of biological samples. And the reason this is important is you can only get brain samples once, but you can get blood samples every week. And that's what we're doing now. We're doing a prospective longitudinal study, collecting blood samples from differentially reared monkeys, starting at birth, and seeing when do these patterns these differences start popping up, and under what environmental circumstances. And finally, with respect to these data, this is not random. They follow particular pathways. So these are the genes involved in the HPA axis pathway that are differentially methylated between a mother and infant. And there is some tissue specificity. For example, all the vasopressin genes you see only in brain, but not in blood, and similarly for the CRH genes. But for many of the cytokines, you see the same pattern in both sets of things. So these, again, are not random patterns. They're following particular pathways. So that's methylation, and as we heard this morning, methylation is not the same as gene expression. Here are gene expression data from another collaboration done with Steve Cole at UCLA. These are collected from monkeys when they're 4 months of age, still in their differential thing. And what you see here, this is a heat plot of gene expression, in this case from leukocytes, white blood cells, and each of these vertical lines is a different DNA point, considered a proxy for a gene. Each of these is a specific subject, mother-reared, 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 peer-reared, 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 peer-reared. If it's red, that gene is overexpressed. If it's green, it's underexpressed, and there's almost no overlap. That is, if it's red in mother-reared monkeys, it's green in peer-reared monkeys. If it's green in mother-reared monkeys, it's red in peer-reared monkeys, with one exception, and that's MR55. MR55 looks like a mother-reared, looks like a peer-reared monkey. MR55 had a neglecting and maltreating mother. And so that monkey looks just like a peer-reared monkey behaviorally, as people at the Hierarchies Primate Center are studying much more formally. So this is gene expression, and again, you get about the same order of magnitude of these early experience effects. Now, I mentioned gene by environment interaction, and this is a different approach. We've taken a candidate gene approach in this, much as a number of biological psychiatrists have done. And here, individual genes have been identified by biological psychiatrists as A, being implicated in emotional regulation, and B, having polymorphisms, that is, more than one version of the gene. And what we do is see if the Rhesus monkeys have the same gene and have the same or similar polymorphism. And the gene that's gotten the most attention in the field over the last 10 years has been the serotonin transporter gene, a gene thought to regulate the reuptake of serotonin presynaptically, the same area that drugs like fluoxetine, Prozac, have their mode of action. And in Rhesus monkeys and humans, there's a polymorphism due to length variation in the number of base pairs in the promoter region of this gene, such that there's a short version of the gene and a long version of the gene. And these are functional differences in that in both humans and in Rhesus monkeys, the short version of the gene is associated with less efficient RNA transcription from DNA, the initial step in the cascade of proteins that leads to gene expression. So does it make a difference whether you have the long version of this gene or the short version of this gene? Well, Peter Lesch, the scientist who was at NIMH in Maryland, and now is back in his native Würzburg, who first described this difference, reported that humans who had the short version of the gene were disproportionately likely to be incarcerated for severe aggression, to be hospitalized for severe depression, or be suicide victims. But not everybody has been able to replicate this finding. It's generated considerable controversy. There is no controversy over the Rhesus monkey results. We've been able to characterize all the monkeys in our colony with respect to this gene. Some have the short version, some have the long version, and some were mother-reared, and some were peer-reared, and we found the results to be interesting. Here's serotonin metabolism, for example. Here, if you're peer-reared and you have the short version of the gene, you have a deficit in serotonin metabolism. If you have the long version and you're peer-reared, you have slightly higher than normal levels. But if you had a good mother, it doesn't make a damn bit of difference which version of the gene you have perfectly normal serotonin metabolism. Here's an example of a specific gene by environment interaction, where I would argue that the interaction is due to buffering effect of good mothering. Good mothering is protecting those infants who have this short version of the gene from developing this kind of deficit. If you look at aggressive behavior, you get the same pattern. Here, if you're peer-reared and you have the short allele, you have very high levels of aggression. But if you have that same short allele and you have a good mother, your levels are lower or as low as individuals carrying the long version of the gene in that independent of rearing. Gene by environment interaction, buffering by good mothering. It's even more dramatic when you look at alcohol consumption. Here, if you have the short version of the gene and you peer-reared, you drink to excess. But if you have that same short version of the gene and you had a good mother, you actually drink less than normal. What may be a genetic risk factor for individuals with adverse early experience may be a genetic protective factor for individuals with good early experience. Complete crossover. And we see the same pattern over and over again with this gene when you look at ACTH levels, when you look at behavioral disruption following separation, when you look at neurobiological development in the first month of life. You see this pattern of if you have adverse early experience and the short allele, you have a problem, but if you have that same short allele with good early experience, you're actually as good or better than normal. And it's not just for this gene. We've now looked at about 8 or 9 other candidate genes, and we get exactly the same pattern. Here's MAOA gene, another gene that's been extensively studied at the human level and associated with aggression, but only in combination with adverse early social experiences. We found the same pattern in monkeys. Neuropeptide Y, the opioid gene, dopamine transporter gene, DRD4, NOS1, BDNF. Here, the same pattern over and over again. Here's data showing that same pattern from Christina Barr's study looking at a CRH SNP. And here the measure is alcohol consumption. Here's the less efficient version of the gene. And when it's associated with peer rearing, you get excessive amounts of alcohol consumption. But that same allele associated with good mothering or competent mothering, you get lower than normal alcohol consumption. So what does this tell us? This tells us, among other things, that good mothering is pretty damn important and go a long way toward protecting individuals who carry these so-called risk alleles from developing the kind of problems that I've been describing. And because good mothering, or mothering style in general, is passed from one generation to the next, much as we've heard described in other species, here's a mechanism by which a so-called risk allele can stay in a population generation after generation with no apparent ill effects. If you have a mother who has a daughter who carries this gene, this allele, but she is a good mother, then that daughter will not only grow up normally, but be a good mother to her own offspring who may carry that allele, and you can see where this can go. On the other hand, if something happens to compromise the ability of a mother to take care of her kids, maybe there's been a death in the family, maybe the old matriarch has died, maybe there's more competition from other families in the group, maybe a new troop has moved into the area and is now causing instability. Maybe there's been a food shortage. Maybe there's been an increase in predation. Maybe you have a disaster like a Hurricane Katrina. Any one of these environmental events that might compromise the ability of a mother to take care of her kids in optimal fashion may put those of her kids who carry these risk alleles at risk for developing the kind of problems that we've done. So we've been looking at early mother-infant interactions very carefully to see what is characterizing these sorts of patterns of good mothering. And I want to show you a couple of videos. The first one is of a two-month-old infant with a low-ranking mother. There's a fight going on to the right of the mother. The mother can't rescue her infant. She doesn't have the social clout. So what does she do? She grabs the infant, grabs the flower, distracts it until the problem is over. Now it's free for the infant is free to go out and explore, and off it goes. And the mother now follows just to make sure everything's okay. That's an example of a really good attachment relationship. Let me show it to you again. There's the infant looking away at the fight. Mother grabs the infant, grabs the flower, distracts the infant. Now the monkeys in the background are running away. And look at that face-to-face interaction. The reason we looked at it is because it's not supposed to happen. Rhesus monkeys are not supposed to engage in face-to-face interaction with their offspring. Human mothers do it, of course, and it's a major part of early mother-infant interaction, but not monkeys. 50 years of research in the wild of mother-infant interactions, and nobody's ever reported it before. Okay, here's a 19-day-old infant. And now not as only face-to-face, there's actually an interaction going on. This is not supposed to happen, the literature tells us. But you just saw it. So when we actually started looking at this, we found it happens quite a bit. Here's mutual, over the first month of life, here's mutual gaze. Red is interactions of infant with others in the group. It's 0 all the way through. But with the mother, it's pretty high. Mutual gaze, lip smacking by the mother to the infant, lip smacking by the infant to the mother, back and forth. It's pretty frequent, 2 or 3 times every 5 minutes. But what happens? At 3 weeks or 4 weeks of age, it disappears. It drops out completely. And what's going on unlike in humans, where of course it keeps up for months, if not years. So what's going on at 3 and 4 months of age? Well, that's the age when infants start leaving their mothers for the first time. And it's probably more important for them to start paying attention to other things in their environment. They've already established an attachment relationship with their mother. They already know that they can run back if they get in trouble. And so so it's probably more worth their while to pay attention to things in the outside. And besides, they have to get that behavior out of their repertoire, because when monkeys get older, the best way to start a fight is to stare somebody in the face. And if they keep doing that when they get older, they'll be starting unnecessary fights with other monkeys. So it's in their interest to stop doing this. And they do. But there's something else worth noting. If you look at these interaction patterns, they're there at birth. They're there at day 1. Now, this will take a second. Sorry. I'm sorry, this is... Okay, here we go. Okay, here's a 3-day-old infant. And the individual on its right is not its mother. It's Pierre Ferrari from Giacomo Rizzolatti's lab in Parma, Italy. Rizzolatti is the guy who discovered mirror neurons 15 years ago. They don't have any infants, and they wanted to see how early you might get mirroring things. So what Pierre is doing with this infant is giving a still face for 30 seconds. Notice how interested the infant is in the face. Who said infants were interested in the face? Now he's going to open his mouth. And watch the infant. infant's opening its mouth. It wasn't doing that before. That sure looks like neonatal imitation. First described by Andrew Metzoff and Tiffany Field 30 years ago. Here's another 3-day-old infant. Now Canvana is going to stick its tongue out, his tongue out. Watch the infant. See that? See that? See that? That's neonatal imitation. Let me play it one more time. Well, I can't do it. So we've been...
You want to play it again?
No, yeah, I'll play it again. At least I think I'll play it again. There we go. The infant starts imitating on the fourth tongue protrusion. Can I have some? Can I have more time? So we've been making faces at baby monkeys quite a bit the last couple of years. And we now know 2 essential facts about it. First of all, like human infants, it disappears very early. In human infants, neonatal imitation is gone after 1 month. In Rhesus monkeys, it's gone after a week. 4-to-1 ratio, right on time. Secondly, not all human infants imitate. In the human studies, the investigators took the non-imitating infants and threw them out of the study because they were messing up their data. Not all monkey infants imitate either, but we're much more interested in the ones that don't. So we're not tossing them out of the study. So here among nursery-reared infants and lip smacking, these are imitators, these are not. Interestingly, when you look at mother-reared infants, and we get to test them just once on day 3 when their mothers are going through their postpartum veterinary examination, but the infants are still on their mother's ventrum, but we can still make faces at them, we find that 95% of those infants successfully imitate, as opposed to 55% in the nursery. So already, by 3 days of age, there's a big difference. There's a big effect of early social experience. Well, does it make a difference whether you imitate or not during that first week of life? Well, we follow these infants up and watch them grow up, and we now know that in the first month, those that imitate are much better at sensory motor integration tasks, like reaching and grasping, taking an object and grasping it. It requires integration of sensory motor capabilities, exactly what mirror neurons are supposed to do. And the ones who imitate are significantly better at this throughout that first month than those that don't. When these monkeys get to the age where they should be starting to play, we find that those that imitated play twice as much as those that failed to do so in that first week, especially during the months where play is really starting to take off as a major part of their repertoire. And perhaps most intriguingly, we found that these infants, by the time they're 2 years of age, are now living in well-established social groups. Those that failed to imitate during that first week of life show much higher levels of self-directed behavior at 2 years, grasping their bodies, sucking their thumbs, than those who did imitate. And most intriguingly, those that failed to imitate by 2 years of age, many of them are developing spontaneous, autistic-like stereotypic patterns of behavior. None of the infants who imitated during that first week show any of these stereotypies. So we just might have stumbled on a very early screen for risk for developing autistic-like stereotypy, at least for the monkeys. Needless to say, my colleague working in the field of autism in humans are starting to look at this as a possible early screening device. So what does this all mean and where is it going? Well, one of the things we're interested in doing is finding out how long and how powerful these long-term effects may be. And secondly, we're trying to see whether it's possible to intervene early on with these non-imitating monkeys by giving them the stimuli that they would be missing by being, if they would be getting if they were with their mother, and seeing if we can get them back on track and whether these longer patterns of problems disappear when they're get back on these early developmental trajectories. And we also want to look at whether we're seeing changes in gene expression. And finally, we want to see what's going on inside the brain. Now, the original mirror neuron studies were done by Rizzolatti with adult monkeys strapped to chairs with electrodes coming out of their heads, and there's no way I'm going to do that with my baby monkeys. But there is another way, and that's EEG, and down the road from NIH is Nathan Fox, my aforementioned friend, who's one of the world's experts in human EEG measurement, and we're using his techniques with a little cap that fits on the monkey's head with electrodes, fits nice and snugly, and then record during periods of imitation and non-imitation. And when we do this, we find that there's a distinctive pattern of EEG during periods of imitation. It's called desynchronization of mu rhythms at low frequencies, and these are the kind of data that we get. These are recorded from the anterior points 3 and 4 and the parietal points 3 and 4. This is the power in this mu rhythm during a non-social stimulus, a rotating disc. So there's no desynchronization, but when the monkeys see a lip smack or a tongue protrusion from a human model, they show this desynchronization. And then when they do the imitation themselves, we see that same pattern of desynchronization. We don't see it during non-imitating periods, and we don't see it in the monkeys who fail to imitate. So perhaps we now have an EEG marker to go along with our behavioral thing that will distinguish imitators from non-imitators and tell us what's going on. The reason this is interesting is while the imitation itself disappears at 1 week of age, social engagement continues as these monkeys get older, and we can look to see whether this particular pattern is associated with this social engagement as it is with human infants, and also for those who fail to imitate, can we get their EEG back to normal with these sorts of interventions. So we are going to be doing this research for some time, and before I finish, if you'll let me, I just want to acknowledge the people who have been instrumental in this research in my laboratory, in particular Anna Kapalkner at the University of Parma. Here's Pierre Ferrari and Giacomo Rizzolatti, Nathan Fox at the University of Maryland, and my dear friend Elisabetta Vizobergi in Rome, who got us interested in the first place. So I want to finish by putting this into a comparative perspective with respect to the gene-by-environment interactions and ultimately epigenetic sorts of things. When we first started out looking at gene-by-environment interactions, we thought they would be widespread among all primates, that this would be a general characteristic. So we started looking at other species, and one of the species we looked at was the Barbary macaque. It's another macaque, same genus as Rhesus, looks sort of like Rhesus, comes from Africa, the only one of the 20-plus macaque species that's indigenous to Africa. You can find them in Tunisia and Algeria and Morocco. They're the monkeys who are on the island of Gibraltar. Except in the wild, they're almost gone. There's only about 4,000 left. They're highly endangered. There's one place or places where they thrive, and these are nature parks. These are nature parks in various places in Europe. There's one on Lake Constance in Germany. There's one in the Alsace of France. There's one near Toulouse. There's one in the Trier region near Trier. They just opened one near Liverpool in the UK. And here, these are all privately run parks, and they're usually about 200 acres of woodlands fenced in with pathways cut through, and inside the parks are 200 plus Barbary macaques. And every day, tourists, thousands of tourists, come to the parks, plop down their 10 euros, pick up bags of popcorn, take their cameras, and wander through the park with the monkeys, giving them popcorn and snapping pictures in their face, young and old alike. If you tried that with Rhesus monkeys, you'd have 20 lawsuits in the first 10 minutes because Rhesus monkeys are way too aggressive from that. They would terrorize the tourists. They'd steal their cameras. They'd bump over the popcorn. They'd bite the tourists. They'd chase them out of the park. Just like they took over a town in northern India a couple years ago, just like they took over the Ministry of Education in New Delhi, just as they required President Obama, when he visited India, to have a special guard to keep the rhesus monkeys away from the president. It wouldn't work with rhesus monkeys. But Barbary macaques are not aggressive. Not only do males take an active part in the rearing of the offspring, so here are two males tossing an infant back and forth. Mother is off screen. Rhesus monkey mother would never stand for that. And when they get into fights, it's all bark and no bite. There's virtually no physical aggression. So we got interested in seeing what their serotonin transporter might look like, given our relationship we found in aggression. So as you may recall, here's rhesus monkeys. And rhesus monkeys have a short version of the serotonin transporter gene and a long version. And so for Barbary macaques, when we looked at them, we couldn't find anybody with a short allele. We couldn't even find anybody with a long allele. Instead, every single Barbary macaque in 7 different colonies that we looked at had an extra long serotonin transporter gene. So then we looked at pigtail macaques. Pigtail macaques are indigenous to Southeast Asia, Thailand and Indonesia. They're bigger than rhesus monkeys, but much less aggressive. They have much higher levels of 5-HIAA than do rhesus. And they also only have one version of the gene. It's a long version of the gene. Then we went to Strasbourg and saw Bernard Terry's beautiful collection of Tonkanen macaques. Tonkanen macaques come from only a tiny part of the island of Sulawesi in Indonesia. No, you can't find them anywhere else. They're beautiful animals. They're twice as big as rhesus monkeys, but much less aggressive. Only the long version of this gene. You can see where this is going. Crab-eating macaques, also from Indonesia. They can only live in rainforest areas. They're smaller than rhesus monkeys. They're more skittish and I think stupider than rhesus monkeys, but they're also less aggressive, and they also only have the long allele. Stumptail macaques, indigenous to India, but also highly endangered. Their ranges overlap with rhesus a little bit, but rhesus can live in a hell of a lot of places that stumptails cannot. They're notoriously peaceful. Frans de Waal once cross-fostered some rhesus monkeys into a stumptail macaque group, and he ended up with really mellow rhesus monkeys until he took them out of that group and put them back with other rhesus monkeys, and then they became as aggressive as ever. Also, only the long version. And finally, we got some blood samples from Tibetan macaques, who are obviously indigenous to Tibet, and the literature for Tibetan macaques from zoo studies said that these were mildly mannered monkeys. But just as we were genotyping them, I got a call from my dear friend Carol Berman, who's probably the world's expert on mother-infant macaque interactions. And she had just come back from a field study in Tibet of these monkeys, commissioned by the Chinese government to see if they might be suitable for nature parks like the Barbary macaques are in Europe. And she said, Steve, I got a problem. I just came back, and these monkeys are supposed to be peaceful and mellow, and they're the most aggressive animals I've ever seen in my life. They made rhesus monkeys look like pansies. They pushed tourists off of mountain trails. I said, Carol, I got a surprise for you. We just genotyped them, and they're all homozygous for an extra short version of this gene. Now, I'm the last person in the world who's a genetic determinist, and I certainly don't believe for a second that a single gene can be associated with so complex a behavior as aggression, but you gotta admit, at the species levels, it seems more than coincidental that you see this relationship between aggression and the length of the serotonin transporter gene. But there's one other thing worth looking at, however, and that's of all of these species, the only one with a polymorphism is rhesus. And when people started looking at other primate species, all the great apes, they've got the gene, but they don't have the polymorphism. They've got one or the other. They looked at baboons, no polymorphism. And not just this gene, many of the other candidate genes for which humans have a polymorphism and rhesus monkeys have a polymorphism, none of these genes have that kind of allelic variation. They're all one or the other. So what's different about rhesus monkeys and humans from these other species? Well, one thing that's different is that they can live anywhere. And maybe, just maybe, one of the secrets to their success, and perhaps ours as well, is not some kind of genetic specialization, but genetic diversity. I want to thank the many people who participated in this research, some of whom are listed here, and I thank you for your time.
Skal vi bare få på oss noen mikrofoner her, sånn at folk kan stille spørsmål til oss, og så skal vi ta spørsmålet videre. Okay, so excuse me, that was very loud. The floor is open for questions and possibly answers from Steve, and also I think at least some of the people who have given talks earlier today are still here. Vi kan også ta spørsmål på norsk, hvis noen føler det er mer bekvemt å stille spørsmålet på norsk, og så skal vi prøve å få oversatt det på en brukbar måte til engelsk. Og siden jeg ikke er biolog, så har jeg da med meg kjære Stig, som tar de mest tekniske biologiske spørsmålene. So. Come on, guys.
Maybe they don't understand my English.
I think they did. There were not that many questions for the other speakers either.
There we go.
Yeah, Christian.
So I guess in one way of looking at epigenetics is that it's just another way of regulating genes. So I was wondering, which really doesn't have a bearing on inheritance or anarchism or anything like that has been discussed today. So I was wondering, have you checked the methylation pattern in the germ cells and connected to that you measured in the blood? So because the blood cells are very different from the brain because the brain is virtually post-mitotic. So how stable are these patterns in the blood if you change the hormone?
So this is exactly why we're doing longitudinal studies. We want to see how stable these patterns are and how sensitive they are to both maturational issues and changes in their physical and social environment. We're also in collaboration with Moshe Zif now looking at placenta, and the next stage will be trying to get at the germ cells as well. But these are the sorts of questions that need to be addressed very early in what's rapidly expanded new field of investigation, and we still don't know the ground rules.
Yeah.
I have a question on another species, but I guess this species also interests you. You've given us a case study where you see that the role of the parental environment, the mother, is very important. And then from other branches of human psychology, we hear more reports that the role of the parents for the future of the kids is over-exaggerated, and it's friends, not the parents, determine the success.
So what I would say is that just like my monkeys, it's both. There are different components to socialization, and the kind of relationship that monkeys form with their mothers are fundamentally different from the kind of relationship that they form with peers. The hallmark of mother-infant relationship are attachment-related sorts of things. The hallmark of play interaction of peer interactions is social play, and you get into trouble if you don't get enough of either. So it's not one is more important than the other. It's that they complement one another under ideal circumstances. What was surprising to us in the last research that I presented was how important those very early initial days and weeks of life appear to be. We always thought that they were not irrelevant, but not really that important because you could always catch up later. But what it suggests instead is some of these patterns are being established right in those very early interactions, and that we ought to be paying more attention to them and promoting them where possible. And sure, later on, those peer relationships will, as infants, both monkey and human, are spending more time away from their mothers and their social world is expanding, then those other relations will take on a role of their own as well. What we're interested, again, going back to the epigenetic story, is are these changes in focus of social activity, are they reflected by changes in either gene expression or methylation patterns as these individuals are growing up? And that's why longitudinal studies in this area are so fundamentally important.
Stig, you have a question yourself?
Yes, I might have one. Is personality polymorphisms become more and more appreciated to be important in ecology and evolution? So people are starting to get this into life history theory and so on. Appreciating the importance actually that you have this diversification of personality profiles. We see that in many animal groups, in fish and other. And my question to you here is that if you think about why they have this genetic polymorphism, and then they get the personality polymorphism as such in a peer situation and in the mother situation. And could it be that there could be an adaptive reason for it? That, for example, when this situation emerged, that they were deprived of the mothers, could be a population phenomenon, so there will be a totally new social structure to relate to, so that it was just not a single individual that was normally deprived, but there could be a population phenomenon that some sort of catastrophe or whatever would work on population, so that you had an adaptive reason for changing. So then you could keep your short allele, and you could compensate for it in a normal situation, and you could release the potential of that allele in a new environmental context.
Yes, and first of all, I think that the term risk allele is not a good one. I think in the case of serotonin transporter, for example, sensitivity allele would be much better. So these individuals carry the short allele, are more sensitive to the bad things in a bad environment and the good things in a good environment. And if you didn't have some circumstances where there were positive outcomes of the so-called risk allele, it wouldn't stay in the gene pool very long. And the notion that there may be whole populations of this, we have some very preliminary evidence, again, for the serotonin transporter gene, that this might be the case. There are some monkeys who have gone over the Himalayas, and their descendants now live in China. But the environment in China is very, very harsh. And the groups are smaller, there's much more aggression, but they survive in places that humans can't. When you look at those monkeys, or at least the ones that end up in lab circumstances in the US that get imported, we find that the incidence, the gene frequency of the serotonin transporter gene, the short allele, whereas it's about 70%, it's about 30% in most Indian-derived colonies, in these guys it's 70%. It's the rule rather than the exception. And when you think about it, maybe it's adaptive to be aggressive and impulsive in situations that are very dangerous and that the resources are very scarce. So behaviors that would pull you through in these situations would maybe get you in trouble under more stable and benign environments. And that's why I think the notion that the idea that weed species are likely to have more of these polymorphisms, because there's no one environment for them. And what might be good in one environment may be maladaptive for another. But if you have that genetic variation, you can move into a new environment, and somebody in the group's going to make it, and then the frequencies will change. So again, it's just an hypothesis at this point. But the notion that this kind of diversity is useful when you are faced with not one environment that's stable, but multiple environments that may be changing, that may be to your advantage to have that kind of variation built into your system.
There's another question.
Do we know if, in the slide that you show with the different species of monkeys, and with the presence or absence of polymorphism, do we know how it is in our closest relatives? Because we know also that chimpanzees, for example, can explore different kinds of habitats. They can live in rainforests, and they can also live in what we call savannas.
Yeah, but they can't live in Oslo. Or they can't live in the desert.
No, they cannot live in the desert, but they can live in a few environments.
Yes.
So my question is, are they polymorphic or not for this?
Not for the serotonin transporter gene, and not for some of the other genes that have been looked at. I won't say all the genes because that investigation is not complete. But in cases where they have looked, the absence of polymorphism in other primate species, even among the great apes, is, to me, very striking. We did not expect that at all. And we did not go out looking for this kind of finding. It just, the more species we looked at, the more clear this pattern became. And I would expect you to find something like that in other taxa. That is, say in rodents, my guess is Norway rat. It's got a lot more polymorphisms than some of its closest relatives, and the canines in the US, the coyote, the same sort of thing. Individuals who can live in multiple places, the argument is that they have to have, that genetic diversity helps.
Okay. Jens?
Yes, I can ask a question more about language rather than reality, because there was people in the beginning today linking epigenetics to evolution. And I'm a bit curious on your view of if we need a more precise definition of evolution after we know more about epigenetics.
Was the question to me?
No,
Or to
no. I want to keep it in English so you can be included.
No.
You
Let
can ask.
Steven take.
He's the theorist.
Just to update Stephen a little bit on, or Steve, I think I'm supposed to call it, on this issue. There was, in some of the talks, there was made a point that one should distinguish between epigenetics as a kind of plasticity of the individual from epigenetics as a question of inheritance.
So I've been hanging out with Michael Meany and Moshe Ziff of the McGill group that basically were one of the first to show epigenetic effects, that is differential maternal licking and grooming by the mother has consequences that show up not only at the level of behavior and biology, but also at the level of gene expression in glucocorticoid receptors in the hippocampus. That was one of the first demonstrations of postmeiotic methylation. And their argument is that it's part of a fine-tuned system that allows an organism or organism and its offspring to adjust to fluctuations in the environment, and that you're likely to see more of this when there are lots of fluctuations and relatively little of it under very stable circumstances. And they, in fact, have demonstrated beautifully that you can have what are called K strategies and R strategies in the same species, because when these rat mothers are in stable environments, they tend to have a much greater likelihood of being high lickers and groomers, with the consequences that carry over to the next generation. But when you change their environment and make it unstable, suddenly they pay less attention to their offspring and become more sexually active. So it's a very nice trade-off, where under stable circumstances there's more parental investment, and so you spend more time with fewer offspring, but that in that same line of individuals, when you change the environment and make it more unstable, suddenly they reproduce more but pay less attention to individual offspring. So you have, we've known that K strategies and R strategies exist for different species, of closely related species, but we never have seen it demonstrated in the same species at different points as a function of environmental stability. And I think that's a phenomenal discovery, and it takes nothing away from traditional patterns of evolution and changes in DNA sequences via mutation, because none of these are affecting DNA sequences. What they are affecting is gene expression of those DNA sequences. And so one provides a long-term stability and preservation of the species, and the other allows for fine-tuning of the characteristics of that species as a function of the place that individuals in that species end up finding themselves growing up in. And both seem to be very nice solutions by nature to solve how species can maintain intact but still survive in changing environments. And this is so new, and I'm not a geneticist, and I'm not an epigeneticist. I have zero training in that area, but I do have colleagues who I think are pretty good. And there's a lot to be done in understanding the relationship among these different patterns of evolution. And Michael Meany describes their findings as being quasi-Lamarckian. They're not true Lamarck, but they also don't involve any changing of DNA sequence. Those are preserved. And I think we have a lot to think about conceptually as to how these things fit together and under what circumstances you would expect to see more of one of these patterns rather than the other.
Hello? Yeah. I think this has been a great day for Lamarck.
Quasi-Lamarckian.
Quasi-Lamarck, but we have also heard that Lamarck was more like quasi-Lamarck than Lamarck. So if there is nobody else who would like to ask any question, I think I will...
It was mentioned in the introduction that you like to think about the intersection between science and politics, and I was just wondering, what kind of perspective do you have on transferring this knowledge into society?
First of all, let me tell you that I'm an employee of the U.S. federal government, and I am under strict instructions from my public information office and his and her superiors to keep my discussion of presentations to the monkeys and let other people come to their own conclusions regarding human implications. And I'm the first to say that rhesus monkeys are not furry little humans with tails, but members of another species with different characteristics. And you have to be very careful in generalizing from one species to another, the real specifics. But I think some general principles emerge from the sorts of work that I do that do have some human applications. And that is the power of social stimuli, how important they are in affecting not only the way we behave, but the way our body behaves and the way our genes are expressed, and also how important those early experiences are. And we're, as a society, I think, put ourselves at risk if we neglect them too much.
I think with that comment, it's a nice place to stop, and I would like to thank Steve so very much and the audience, but I would like also to give the last word to Kirsten, who has done so much for this conference.
Thank you. I will talk in Norwegian. Det flere elever fra Fagerborg igjen? Har dere gått alle sammen? For nå tenkte jeg skulle si at nå har vi skjønt at epigenetikk omtrent som biologi. Det spenner fra molekyler til, ikke minst, Stigs matematikk, filosofi og psykologi, og ikke minst replikasjonsrate-maksimeringsprinsippet fra Bergen. Og det betyr at selv om vi en gang, kanskje det var i januar, bestemte oss for at epigenetikk årets konferanse, no doubt about it, så var det litt å ta tak i. Og i den forbindelse så takker jeg Ivar Mysterud, for han kom springende med boka til Jalonka og Lamb, Evolution in 4 dimensjoner. Og da hadde vi et byggverk rett og slett, og det var å putte inn og finne foredragsholdere. Og med det vil jeg jo takke foredragsholderne våre i dag. Det har vært helt fantastisk. The last one is Suomi. And to Suomi, we need to give you a gift for your talk. And this is a red blanket of University of Oslo. You need to take it with you home to remember us. And we hope you will come back to Norway. You're here in November and it's even sunny.
Yes, I brought it with me.
I know. And til foredragsholderne som også har fått en hagebok, så tenker vi også at vi skal bekranse dere. Og nå ser jeg at mange av dere har vel gått, men noen her. Men jeg har ikke tid til å ta alle sammen ned. Kjetil sitter der for eksempel, Stig sitter der. Denne minnepinnen, den tror jeg rimelig stor, så dere kan ha glede av den. Jeg vil at dere skal merke den med Biokonferansen. Det betyr at vi kan komme igjen. Og på den minnepinnen så vil jeg også trekke frem den traileren, som vi har kalt det, som Norit har jobbet. Det mye arbeid, og det ligger veldig mye informasjon om hver foredragsholder og om Biokonferansen på den traileren. Så den ligger på pinnen. Og det et minne fra i år, men som sagt, pinnen kan brukes neste år når dere kommer her litt i siste liten og putter inn pinnen før PowerPointen skal opp. Så tusen takk til Marit og Norit. Kjempejobb. Jeg synes de fortjener en kjempeapplaus. Jeg har dratt hele... Og så foreslår jeg da bekransning av foredragsholderne, sånn ettersom at for eksempel Maria kan begynne å dele ut. Når det gjelder det som skal takke, så tenker jeg nevner ikke flere navn, men seminaret i vitenskapsteori ved Camilla Serkansen. Tusen takk. Realfagsbiblioteket og Fritt Ord ved Live Rasmussen. Vi hadde ikke fått denne biokonferansen, hadde det ikke vært for dere. Og ikke minst Torborg, daglig leder av Bio, som har bundet det sammen og som til de grader har holdt tråden og kontakten med alle sammen. Og til dere som kom, tusen takk. Jeg håper at dere har fått mye ut av dagen. Det har i hvert fall jeg selv fått. Det slik da at vi kan møtes nå til mingling på godt norsk. We are going to have a glass, a drink, some tapas. And for those of you, eller dere, Suomi, are going for dinner in the restaurant Tabibito. And I suggest that you follow Maria. And that means vi møtes i biokantinen nå for å få litt tid til å snakke sammen. Og der kan man bare forbli hvis ikke man har meldt seg på at vi felles reiser klokken 16:30 fra hoveddøren på Kristine Bonnevis hus. Og der kjenner dere igjen Maria sitter der på venstre side av meg. Kan vinke. Sånn at vi felles avgang fra Blindern ca. 16:30. Og da møtes vi på restaurant i byen. Ha en riktig god kveld, og takk for oss.